Thanks very much again, Paulina, particularly for inviting me. I'm going to talk about using systems thinking to approach chronic disease treatment with multiple sclerosis as an example. And if you're familiar with the great work of Ivor Cummins, he's done something very similar with particularly heart disease or cardiovascular disease. And it's worked very well, I think. So everybody knows all this, we did it the other day, but I did a degree in biochemistry, then a medical degree, then I ran around the halls of the hospital working as a doctor in London for a couple of years, and now I'm doing a PhD looking at optimizing resuscitation after perinatal asphyxia. And actually we've had some really interesting conversations about why, if you use an ancestral approach and look at sort of evolutionary development, you can kind of see where we're going wrong in terms of resuscitating babies if they have problems during childbirth. In my spare time, I write a blog and have a podcast with my friend Chloe, and then I do some diet and lifestyle and rowing and strength and conditioning coaching on the side, just as a hobby. But this project, Systems Analysis in Multiple Sclerosis, is something I've been doing for about 4 years. It's an international multi-center family project that I've done with my mum, who's at the University of Iceland, and my stepfather and stepbrother, who are both at the University of Lund in Sweden. And we presented this work at the Systems Dynamics Society in Switzerland and the International Society for Neurovascular Disease in Poland. We have a few patients that have heard about our work that we work with, obviously in concert with their normal neurologist. But the important thing is that the whole idea is that this is a toolkit that we can use in any complex disease. So what is systems analysis? This is basically something that engineers, particularly chemical engineers, which my stepfather is, are trained in. It's just taking any big system, complex system, finding out all the individual parts, how they interact with each other, and then in medicine, hopefully we can sort of interpret the big picture, find some root causes to do a root cause analysis, and then potentially come up with some treatments that we didn't expect originally. And we go through this process even when we're designing a model. So we'll have a model, we'll have some knowledge, we'll test the model against our knowledge, and then maybe we'll have to redefine the problem, ask some more questions, go and find out some more information. And through an iterative process, we'll kind of get a model of how we think the disease works. So we do this with mind maps and flowcharts, and then we create things called causal loop diagrams. So this is an example here. And what's nice is the fact that, as well as the traditional cause and effect that we might expect, or we talk about a lot in medicine, using systems dynamics and systems analysis, there might be other effects that we didn't expect, and these can then feed into the system. So it gives us a much bigger idea of what's going on. And you'll notice that we create two kinds of loops. One will be a reinforcing loop, so it's kind of like a feed-forward effect, or a balancing loop, so we'll have a feedback effect like you'd expect in any sort of physiological system. And this is basically just talking about how we do it. So the first half is systems analysis, where we'll try and create a model, and then the second half is using systems dynamics to put that model into a computer and run with it and see whether it gives us some useful answers. And so the question is, why do we need systems analysis? And I think at the moment, we talked a lot about how traditional allopathic medicine is really, is failing us. We're doing all this research, all this money is going into it, and it's not really getting us anywhere. And I think part of that reason is because we're just thinking linearly. So it's just cause and effect, and then the downstream effects aren't really thought about. But this ignores basic physiological concepts like homeostasis, adaptation, particularly in terms of metabolism, and then also just those basic things you learn when you're learning physiology, negative and positive feedback. And somebody thinks you can intervene into a system and then not expect any downstream effects, and that's just not what we see. And we don't know everything, we can't expect to know everything, and we're learning everything new all the time. And what's good about systems analysis is you can see holes in your model, you can see holes in the system, and then you know where you need to dig a little bit deeper. So some examples, think of the failure of linear thinking in allopathic medicine. So single molecular targets after stroke. There's a really nice review in the Annals of Neurology in 2006 where they talked about over 1,000 experimental treatments that have worked in cell culture, worked in animals, and have no effect on stroke in humans. And these are all single molecular targets that affect one pathway in one cell type, and then they expect that this is going to actually have any effect on the multiple pathways and multiple cell types over multiple different time ranges that happens after an acute stroke. And then again, something like the calories in, calories out model of obesity, we know that we reduce calories or we increase output, you won't get sustained fat loss or metabolic health. So there's got to be something more complicated going on. But then as I look at the sort of the ancestral health community and not people in this room, but in a greater context, people are still going down the same road. You know, everybody has a favorite angle, so they talk about the gut or the brain or the adrenals, or they'll talk about just one macronutrient. There was I recently took part in an online conversation about the potential benefits of low carbohydrate diets, and a very famous sort of paleo antagonist weighed in and said that none of the low carbohydrate trials mean anything because they didn't control for protein intake. And obviously protein intake is important, but as soon as you start making arguments like that, you really lose some of the benefit, and you're not having a useful conversation. And again, we see a lot of people who are basically blaming carbs on all disease, and obviously carbohydrate restriction can treat a lot of disease or help treat a lot of diseases and may mitigate some of the other things going on, but I think that we can't just focus on one particular thing, we have to look at the whole system. And so I think it's important to remember that the inverse of the treatment does not necessarily mean the cause. And again, so like metabolic health and carbohydrate restriction does not mean that carbs are the reason why the person got fat in the first place, and I think there's a lot more going on. So if we look at the way that doctors think about multiple sclerosis at the moment, there's a lot of question marks, they're not really sure what's going on. There's definitely some problem with the blood-brain barrier. There's an immune component, definitely. Hopefully the body's trying to repair itself, but eventually you'll get some kind of neurological dysfunction which will lead to disability. And when I look at this, and when we first came up with this, it reminded me of this, which is kind of how most people tend to think about metabolism. And as we know, it might just be a little bit more complicated than that.
Is that why you wanted it so close so we can see that?
Yeah, I want you to talk about this. So if we look at the current therapies in multiple sclerosis, they tend to focus around T-cell inhibition and immune modulation. And these might be things like infliximab, so TNF-alpha inhibitor, Gilenya or fingolimod, which is a sphingosine-1 phosphate receptor modulator, and basically sequesters T-cells within the lymph nodes and stops them getting out, or something like Tysabri, which prevents T-cells crossing the blood-brain barrier and getting into the central nervous system. And we know that T-cells are part of the pathological process in multiple sclerosis, but these do not prevent final disease progression, and though they do reduce, they can reduce MRI changes, they can reduce relapses and remitting MS, those things don't predict final disease progression. So I think what's happening is we're ignoring the initial insult. And what's happening is, I think, is very similar to when we talk about LDL, as the fireman at the scene of the fire, and these T cells are getting involved because of some damage that's already occurred, and we're blaming them for the problem. And I think it's just part of shooting the messenger. And we know that T cells are critically important in terms of neurological repair. In animal models, if you do something like an optic crush model, and then you remove T cells that react against myelin basic protein, you actually get a worse injury and you don't get any reparative processes. So the way that we're treating currently, we do maybe get some improvement in quality of life, but we're not preventing disease progression, and we're not allowing for repair. So what we did, we basically went into PubMed, found as much information as we could. We got over 1,000 references, but also case reports. So Roger McDougall was somebody working with the paleo-style diet back in the 80s and 90s in patients with multiple sclerosis. Roy Swank was working in the 50s and 60s. It was actually more like a low-fat, plant-based diet in patients with MS, and both of them were seeing some results. So we basically took all of this, we assumed that nothing was important or more important than anything else, and everything is potentially relevant. And we found some core components. So obviously the immune system is important, vascular flow dynamics can be important. We're damaging the blood-brain barrier, there's a balance between oxidants and antioxidants. At some point, there's a loss of self-tolerance, and that can occur through molecular mimicry. This can be through diet or infectious causes. Environmental toxins may be causing initial damage, so heavy metals, EMF, endocrine disruptors, and then some things can kind of keep the system going, and these often tend to be lifestyle factors, and we want to try and boost any regenerative processes that could be going on. So we'll take each of one of these little pieces and we'll create a causal loop diagram, we'll create a mini system, and then we'll see how those systems interact, and hopefully we'll get kind of a bit of a bigger picture and distill some useful information out. So this is a simplified version of infectious processes that could be happening. So Epstein-Barr virus has a duplicitous role. We know that if you're infected with it very early on, it can be protective against multiple sclerosis, but if you're infected later in life, then it can increase your risk. Then there are also things like Pseudomonas infections, and they can work through two pathways. One might be through molecular mimicry, so antibodies against Pseudomonas can then also cross-react with antigens in the central nervous system, but they can also cause biofilms, which can affect the blood-brain barrier. And what we also know about viral infections is that they can change the Th1 to Th2 balance of the immune system, and you get more of a cytotoxic effect, which can then worsen disease progression in multiple sclerosis. And the next slide is about venous insufficiency in multiple sclerosis, and Josh will probably know that this is quite, this is a very contentious part of multiple sclerosis etiology, and this goes back to 150 years ago, when Charcot was first describing multiple sclerosis. He noticed venous vascular abnormalities around or near the lesions on autopsy. And recently, there are some big proponents who say that a vascular cause is the main cause of multiple sclerosis. But one sort of the first randomized controlled trial that came out last year said that there was no benefit to improving vascular flow in multiple sclerosis patients. So I think we haven't entirely figured this out. It might be important, it might not. But what the important thing that I think is that what they're doing is they're addressing the root cause. And the root cause is that if you have some kind of vascular flow problem, much like if you have venous incompetence in the legs, you get hemosiderin deposits around the blood-brain barrier. And then through an oxygen cascade and the Fenton cycle, you then can damage the blood-brain barrier, damage myelin neurons and central nervous system locally. And then once you're in, this will kind of self-propagate, and that can give you the initial insult into the disease. So it's a potential root cause. And then we start to look. I know it's complicated, but I'll talk you through it. And this is where the effect of diet can come into multiple sclerosis. So there's a really nice paper by Dadis Karazian and his colleagues that came out a couple of years ago, and they looked at the fact that antibodies in the blood that react with particularly wheat and dairy proteins, they can also cross-react with myelin basic protein or Purkinje cells in the cerebellum. So antibodies against these kind of dietary factors can then go on and cross-react in the central nervous system. And then, I did most of this work 2 or 3 years ago, and then I spent a lot of time working in hospitals, and then didn't really look at it, and then I came back, and then when I came back, I noticed all this stuff that we started talking about in the ancestral health community has really sort of picked up. So things like the omega-6 and omega-3 balance can affect local inflammation, and then things like anthocyanidins in coloured vegetables, berries particularly, they can actually inhibit matrix metalloproteases, which contribute to blood-brain barrier damage. So actually, just if you've got a leaky gut, you've got a leaky blood-brain barrier, you eat a nice load of blueberries, and you're actually delivering these inhibitors right where you want them to be, and that can actually improve the integrity of the blood-brain barrier. And then there's other things like if you have an A1 beta-casein in your milk, you have A1 cows, then beta-casomorphin 7, which is generated, can directly degranulate mast cells, and that will contribute to your inflammatory processes as well. And then over here, this is something again we've talked about recently. This is a very simplified version of all your methylation processes. So obviously your methionine glycine balance, B12, folate, and homocysteine, and these all feed into your inflammatory processes as well as your DNA processes. Balance of omega-3 and omega-6s, but then also things like your selenoenzymes, superoxide dismutase, and these can be affected by antioxidants and fat-soluble vitamins. And then something like sugar intake can increase inflammation both within the mitochondria, but through its action through complex 1 of the electron transport chain, or just something like the glycation processes that we were talking about yesterday. And this is just another bit of that. Obviously, tocopherols are very important. These can be regenerated through vitamin C or alpha lipoic acid. And then we talk about something like Q10 and the effect on mitochondria. And one of the big things that we see, some of the symptoms we see in multiple sclerosis, are because of failing mitochondria. And then, you know, you have a patient multiple sclerosis to get into their 60s, you put them on a statin, and then you've really sort of, you've given them a double whammy effect there. So then, as you start to put these things together, what you'll see is you can see where some of these symptoms come from. So, say you have had some kind of vascular problem, you've got iron, you know, inside the nervous system where it shouldn't be. And then as you're getting oxidative damage, you'll damage things like the astroglia, which support your neurons. And then two things can happen. So you'll end up with neural dysfunction, you'll end up with the motor symptoms or the sensory symptoms you see in multiple sclerosis. But then also you'll affect signaling between neurons, and you'll deplete some neurotransmitters, and then you'll get some of the depressive and mood problems that we tend to see in multiple sclerosis. So you can kind of see how all these systems interact, and then how that leads to the symptoms that we actually see in the patient. So hopefully you'll see that all these groups and systems can physically interact, and then we can actually put it into what turns out to be a very simple system. Yeah, easy. But what's nice about this is that we can create this and then hopefully work our way back so that we have something useful that we can do with patients.
Whoa.
You don't have it in memory yet?
Sending that to Michelle.
So you don't need, so the only thing, this line here is the blood-brain barrier, so this is peripheral and central things that occur. So then we can distill this down, and then we start to look for root causes, both common factors and potential intervention points in patients. And so this is a simplified version. You'll see that sort of here's the dietary components, here's the infectious components, you know, vascular components, and then hopefully, I think the important thing to see here is this is the whole of the immune system that we've distilled down to one point. But if you see all these reinforcing loops here, so compromised blood-brain barrier and oxidative processes there, and neuronal damage, if you remove this, so say you just knock out all the T cells, you'll see that these loops and these systems will still keep going. So if you just take out that one drug there, it won't have any effect on the long-term disease process. And that's the really powerful thing, is we can see that if we just intervene here, we won't have that much effect on the disease. So then we start to look at key factors, and I think the initial insult is key, and that's what we don't look at in terms of treatment, and that's also what we don't look at when we're investigating multiple sclerosis in the lab, because we use a model, something like experimental autoimmune encephalitis, and what we do is we start by injecting a mouse with myelin basic protein, and it starts to react to that, but nothing has addressed the fact that in the human, in the patient, something has caused the blood-brain barrier damage, and something has damaged something locally, so then the immune system starts to respond. And this could be an infectious process, either by affecting Th1 or Th2 balance, or we see things like biofilms in the vasculature with something like a Pseudomonas infection, and that might cause the problem. Impaired vascular dynamics. The environment is obviously very important. And we talk a lot about vitamin D in multiple sclerosis, but one of the things we've started to see now is that there's an interaction between microinflammation in the skin and local vitamin D production, and that increases T regulatory cells centrally. So actually just taking vitamin D as a pill and not getting it from the sunshine doesn't have the same effect. And obviously things, toxins, maybe like mercury, amalgam and fillings. And then there's some kind of genetic interaction, and there's a Norwegian group that's just recently published a paper talking about over 150 different genetic polymorphisms that can predispose people to multiple sclerosis or associated with multiple sclerosis. But when I asked them whether they knew these were directly inherited factors that were fixed, or whether these were just epigenetic expressions of the environment that the patient's been exposed to, they just don't know the answer to that. So this is something that we're really working on. And then things will tend to propagate the original insult, and I think that's where diet and lifestyle really seem to come in. And then you want to try and boost any regenerative processes as possible. So we came up with what we called a sequential treatment approach, and although we didn't start out looking for it, what it looks like is something, it's a very sort of paleo-style diet or lifestyle. And you know, if you're worried about, you know, insurance or things like that, what you can do over here, every patient can avoid gluten, dairy, and legumes. You know, that's free. And I don't think that we have the evidence to suggest that these are the things that are causing multiple sclerosis in the first place, but we have enough evidence to suggest that they could really propagate the disease once it's gotten going. And then we want to do something like boost antioxidant capacity, so anthocyanidins I talked about, which can help blood-brain barrier repair, EPA, omega-3, and then we can boost things like intracellular glutathione by taking isothiocyanates from cruciferous vegetables, which Terry Bowles talks about a lot, by upregulating NRF2. And then we can start to get into diagnostics. And I say that venous ultrasound and MRI angiography is in progress because this is still something that's very contentious and may or may not turn out to be important. But then allergy assessment and then also things like comprehensive infection testing I think is very important. And there's actually a guy down the road in Scottsdale called Stephen Fry who does some very cool like blood biofilm testing with some good effects. And then we want to try and terminate initiators, so anything we found diagnostically we need to intervene with. Immunomanipulative therapy can be important if you've got a very severe immune response happening at the time you need to stop it so that you can start repair. And then regeneration, so things like DHA, proper thyroid function, they're really critical for neural development and for synapse development and neuronal plasticity. Stress and sleep, Deborah just said that if you don't fix sleep, you can't fix anything. And then something like ketones, we know they're very neuroprotective, and that could be something, you know, a cyclical ketogenic diet, or you can just boost ketones by taking something like an MCT oil. And then this, you know, from that big picture that we got, this is what we kind of distill out. So this is the whole system in the middle, and then you see where where we can intervene. So that could be through diet, antibiotic treatment, supplementation. Maybe you want vascular intervention if you've identified a real problem in your venous system. And if some of this sounds familiar, then like I said, this is very similar to what Terry Walls does, particularly the dietary part of it. And she developed this completely separately around the same time through her own work, and I think it's quite nice that two different sets of people do completely different things and they come up with roughly the same answer, and I think that's very powerful. And she's obviously trying that out in trials now.
There was no exercise in your diagrams.
No, so they're the...
well, not that I saw.
No, there is...
Tommy, come on.
Yeah, I'm sorry. There isn't in this one. I've given you the wrong diagram. There is in one of the diagrams. It's important. And that's very important for neuronal programming, and that's something that because some of those diagrams were a little bit old, we just haven't integrated all the latest data. And so then the next step that what we want to do is to then take that model, put it into the computer, and see if we can get something useful out of it.
So that was done by hand?
No, so this is done by, I am not a systems dynamics expert. My stepdad is a systems dynamics expert, and I'll show you the model, and he did that all, you know, you input each of the bits separately. And through this we can hopefully reconstruct pathological histories, investigate some hypotheses, but then we'll also see why just taking out one aspect doesn't treat the disease, like I talked about earlier, and then hopefully explore potential proposed cure. So this is a very basic version of the model that we have, and you don't need to understand any of it. But basically works by you have pools of things. So you might have a pool of like an oligodendrocyte precursor, and then or a pool of intracellular antioxidants, and then you can see how these change as these things interact. And then the nice thing we've been able to do is we've been able to look back and retrospectively model the disease progression in actual patients. So what we tend to see is that relapses and remissions seem to happen in line with increased blood-brain barrier permeability. And so you'll have relapse and remission, and then eventually you'll end up going towards secondary progression.
And can you identify then what switches relapsing remittent to secondary prevention by looking at that graph?
Yeah, so what you see here, so you see like the red line is basically T cell activity. The pink line here is oxidative stress, and this is oligodendrocyte precursor. So it's basically your ability to regenerate. And so what happens is that as your antioxidant capacity drops, or your oxidative stress increases, you'll see this same kind of drop in oligodendrocyte precursors, and that's when you'll switch between the two because you're no longer able to repair after each cycle of damage. And this is what you tend to see here. So over time, this is our oligodendrocyte pool, and then this is things like total brain and other brain structures, and we see sort of total brain atrophy over time, and we can model that as well. So the take-home message, I think, is that this is actually a way that we can approach any chronic disease, and what I really want to do is I want to take all of Bob's data that he put together on the lipid stuff, and I think this would be a really nice way to see how those things potentially interact. And each of those arrows that I showed in those diagrams is, one person or many people or many groups working really hard to identify one part of the problem, and I think that's very important. But then all this data and information is out there, and then if we take a step back, then maybe we can find some answers that we didn't have before. And the important thing is, if you're going to take it to your patients, you can modify the individual risk factors. So you can do the diagnostics, you can see what was probably part of the issue, and then you can also explain, using the diagrams, why one change isn't enough, why you can't just give one drug. And we won't pretend that we know everything. Like we talked about, there's some data that still needs to go in, and we still need to work on the model, and the more the merrier, absolutely. And if anybody wants to learn systems analysis or systems dynamics, they can get in contact with us, and in Iceland, we'll gladly show them how to do it. And then this is just to remind us that it's never just one thing.
Typing this in, she's already got one recruit already.
Right.
Wait, in Iceland?
At
In
HIS.S.
Yeah, at hi.is.
Part-time Iceland. What is that?
It's Halsbull Island, so that's
Can you believe if I
University of
live
Iceland.
in the sun and you live in the North Pole?
Yeah.
Okay, and then any questions?
Will you come next year?
Yeah, of course.
Does the model account for the latitude gradient at all?
So it is in there, yeah,
Okay.
because of but that's we think partially vitamin D and sun exposure, but then also there's things like EBV is endemic in Africa, and we that might be protective, but then obviously you don't get exposed to it in the Western world until you start fooling around with other kids and when you're a teenager, and so
Right.
that's where.
Because I see, I virtually see no white women who grew up in the South with MS, but I have tons of African American, so I assumed that was likely vitamin D related.
Yeah, that's a huge part of it. Yeah.
So
So latitude is very important.
right. Were you in the analyses? Were these mainly, or were these all relapsing remitting patients? Or
Yeah, so I mean, I can if people we have one. So we've done it mainly with relapsing remitting, but we have one patient we work with who had fulminant MS. So she went from fully healthy to in a coma within 2 weeks, and so what they had to do is they were taking daily MRIs, and they could actually see the white matter damage expanding daily, and they basically just filled her with corticosteroids, and then because if you have fulminant MS, you're not in any position to be treated with the traditional sort of immunomodulators. They basically just put her in a care home and left her there. And the nurse looking after her had heard about the work, contacted my stepdad, and we just did some very basic stuff, and we just completely changed the diet and changed what they were feeding her through her peg. And 2 years later, she's able to sort of stand up and talk, although she still has a tracheostomy, and is able to sort of wash her face herself and things like that. So, and she hasn't been taking any drugs. It's just whatever we've been able to do, particularly with diet modification.
And any ideas? So, with MS population, we have about 90% of relapsing remitting that do fine, no matter what I can prescribe them something or not. And then you have like small percentage that eventually will transition to progressive and so forth.
Yeah.
Any idea what's behind that?
I don't think you can. I think the only thing we figured out is that you can't generalize. You have to. There will be something going on, and it could be. For some people, it could be a dietary thing. It could be an infectious process, and we talk about so much about gut health and systemic infections, and I think you just won't. It could. That's where the diet-gene interaction happens. So maybe some people they could still eat the same crappy diet and they don't get any effect from it, and some people then see a problem. So we don't know.
One of my concerns has been that it's obviously a heterogeneous disease to begin with, and is it the same thing? You know.
Oh yeah. So I mean that's impossible. So obviously, you're right. Well, we haven't really worked with anybody with primary progressive MS, and these could all be very. And I think it is heterogeneous, and that's because from what we can see so far, the etiology is very heterogeneous. So I think it's a different expression of different things.
Right.
I have a question. I was really curious about your thoughts about A1 milk.
Yeah.
Is everybody here familiar with A1? It's an allele that was introduced about 5,000 years ago in China. And it's about 95% of the milk on the shelf right now is visible in this
So
country,
it's in the US and the UK, but in Iceland and France they have mainly the A2, and all of the traditional populations, so Asia and Africa, they have mainly A2 milk.
And also goat is A2, we're A2.
Jersey and
Just about
Guernsey cows are
every
A2.
mammal is A2. So I know that it's associated, not necessarily cause and effect with with promotion or worsening of autism and schizophrenia. Is MS now on that list? Or no?
So I think the really important thing there is the fact that when you look at first people didn't think that we could absorb beta-casomorphin 7 and it wouldn't have any effect, just like the gliadorphins from wheat. But we've now seen, particularly in children, that they do absorb beta-casomorphin 7. And there are a lot of people who have it in their blood and don't have any issues. And I think the real difference there, actually, it's not the beta-casomorphin 7 necessarily. It's the fact that they can't upregulate dipeptidyl peptidase 4, which is what then breaks it down. And there's an interesting thing there, again, because dipeptidyl peptidase 4 breaks down the incretins, which are very important for metabolic health. So if you have higher dipeptidyl peptidase 4, you might be protected against ADHD and autism potentially if beta-casomorphin 7 is having a thing there, but then you might increase your risk of later metabolic disease. So we're still really figuring that out. I think just the basic data on beta-casomorphin 7 and just absorption and the effects, most of it's correlative. We don't really have any causation data yet, so it's just kind of theory.
I was going to say that I think basically what systems analysis is in this situation is holistic medicine.
Yeah, no, absolutely. Yeah.
I think it's a great way to brand.
Yeah.
I think that's really what we're trying to advocate for. Right?
Yeah.
The functional
And it just
medicine doctors would say that's what they practice.
Yeah, and all it does is it just gives you a diagram of why it works.
Right. Right. But that's a good way to brand what you're doing in a scientific way.
Yeah.
That you do with scientific background.
And I think the important thing is then that because at the moment we have all these things that we know are really important, and you can sort of work your way through lists.
Right.
But in this kind of you can sort of maybe see how things interact, and it makes it a little bit easier to get your head around it.
And you realize this is not something you can work through in your own head.
You need a model. No, absolutely, yeah.
You brought up cardiovascular disease, and it's so striking the difference between like high cholesterol causes heart attacks,
right?
Which is this like very basic model that
can't be
we've grown up with for 30 years, and it's so obvious that there's this huge complex system that while MS is a terrible disease, and it's like imagine what we could if you did have something like this for cardiovascular disease.
Completely agree.
That would be amazing.
So is there a systems theory for dummies book that would just I don't want to really learn how to do that.
So Harold has written some literature, and so if I have both the basic abstract version published, so like a 5,000 word abstract, but then also the 200-page in-depth version. And if people want them, just bring me a USB stick, and I'll gladly give it to you. And that explains both the process and then what we came up with. But then if you contact me or you contact Harold, he will send you some basic literature on just how to do systems.
What methodology is used?
What do you mean?
System analysis. That's no training. Do you know what methodology is used?
What do you mean?
So there are different methodologies for system analysis. It could be sequential or iterative.
So we basically did it as an iterative process with multiple people providing multiple, through multiple versions. So we basically, it was I'm the biochemistry geek, not the systems analysis expert, but we go back, we did it basically like this. So it's like that. We start with a very basic model, add knowledge. It was an iterative process. Was how we did it. And that's if you want the literature, Darrow explains it in more detail.
Because obviously from a software engineering background, that's extremely interesting. So I'd be interested in what methodology.
Yeah. I mean, it's basically just putting together an algorithm.
Yeah, exactly. So
That was interesting to me too, what you said about having that kind of background, because I think at some point the human brain is just not going to carry all that.
Yeah.
There's got to be enough.
And we've missed so much out of those models. You know, there's so much stuff that we won't pretend we've got in there. You know, it's still very basic, and you know, you're reaching the limit of what one brain can put in one place. Yeah.
So, as you know, I've been doing functional medicine since the late '90s, and this kind of lecture, I suppose you know that, but this is exactly the kind of thing that is presented in functional medicine.
Yeah.
Right.
That complex set of things that lead to health or non-health.
Yeah.
And it actually does simplify down to basic concepts in terms of coming to health.
Yeah.
For example, like doing a gastrointestinal function test, you might see in 1 out of 20 people a high Pseudomonas. And that might have a different outcome depending on their genetics, epigenetics, lifestyle, etc.
Yeah.
That Pseudomonas.
Yeah, absolutely.
And then you can figure out strategies for eradicating that Pseudomonas, right?
Yeah.
With eating differently, with digesting your food better, with the right probiotic, you know, those kinds of approaches.
Yeah.
But there, you know, your lecture reminded me of a systems biology really good article that was published this year by a guy by the name of Dale Bredesen at UCLA. He's in the UCLA Brain Research Center. And he did a study of 10 patients with cognitive decline.
Oh yeah, and he looked at each individual risk factor and he modified what. Yeah,
so
it was great.
So he has a table of 20 risk factors, and he's got the research that documents each one of those risk factors. The whole point of it being, you're not. It's the exact opposite of the one drug for one
size fits all.
thing. It's an approach where we're taking all these factors that research has shown they're associated with cognitive decline. We're not going to fix one. We're going to optimize each of these factors. Right?
That was a great. I saw that paper. I sent it to everybody who I thought would care about it.
Me too, yes.
I think the one.
Dale Bredesen.
The one thing is that it doesn't.
Brilliant paper.
Just where he could move forward is you could see how, you know, if you can map how those things interact, then you can maybe see what's more important, what, you know, any other things that feed in. So like lists are I like the way that if you create a system, you can see how things interact better.
And in different people, different ones of those factors are going to be more important.
Absolutely.
Like 3 or 4 of those factors are going to be important in one person. I mean, and he starts with diet, right? And he doesn't say paleo diet. He says low glycemic diet, right? Which is the more, you know, generally accepted format. Anyway, of those people, all 10 had stopped working, and 6 out of 10 went back to work after working with him in this, you know, multi-dimensional protocol. So just really interesting.
The only thing I was going to comment on is because I love this approach, but somebody has to be the guardian of the schema, because as we learn more, some of these things may fall out.
Yeah, absolutely. Yeah.
So, you know, it's going to be one of those things we're going to have to be kind of conscious that.
And you've got to make sure that people's biases don't start to creep in, because that's, you know, it's really easy to just focus on the. Yeah, you can just keep focusing on the bits that you really like. You know, so we can just, you know, we can just talk about this all day, but then you ignore the other bits, and that's absolutely, that's very important.
Yeah, if the answer is statin time after time, you know.
Or if the answer is gluten.
I mean, they're just not giving enough statins. I think that's.
Thank you.